Gene polymorphism in disease biology. A gene is said to be polymorphic if more than one allele occupies that gene's locus within a population. Gene polymorphisms can occur in any region of the genome. Majority of them are silent and don't alter the function or the expression of a gene. A polymorphic variant of a gene can lead to abnormal expression by production of abnormal form of the protein. These abnormalities may cause diseases in the human body. Contents of the manual: DNA extraction, protocol for gel electrophoresis, protocol for PCR, protocol for restriction enzyme digestion. DNA extraction. Pipette 20 microliters Qiagen protease K into a 1.5 milliliters microcentrifuge tube. Add 200 microliters whole blood sample. Add 200 microliters lysis buffer. Mix thoroughly by vortexing. Incubate at 56 degrees Celsius for 10 minutes. Briefly centrifuge the 1.5 milliliters microcentrifuge tube to remove drops from the lid. Add 200 microliters of ethanol. Mix thoroughly by vortexing and briefly centrifuge the tube to remove drops from the lid. Pipette the mixture onto the CHAMP mini spin column and centrifuge for 1 minute. Discard the flow-through and collection tube. Place the QIA-AMP mini spin column in a new 2 ml collection tube and add 500 microliters assay buffer, AW1 of QIAGEN. Centrifuge at 6000 xg, 8000 revolutions per minute, for 1 minute. Discard the flow-through collection tube. Place the QIA-AMP mini spin column in a new 2 ml collection tube and add 500 microliters assay buffer AW2. Centrifuge at full speed, 20,000 xg, 14,000 revolutions per minute, for 3 minutes. Discard the flow-through and collection tube. Place the QIA-AMP mini spin column in a new 2 ml collection tube, normal microcentrifuge tube, and centrifuge at full speed, 14,000 revolutions per minute, for 1 minute. This eliminates the chance of possible buffer AW2 carryover. Place the QIA-AMP mini spin column in a new 1.5 ml microcentrifuge tube. Add 1/200ths of a microliter elution buffer A and incubate at room temperature, 15 to 25 degrees Celsius, for 1 minute. Centrifuge at 6000 xg, 8000 revolutions per minute, for 1 minute to elute the DNA. Gel electrophoresis. Reagents: TAE, tris, acetic acid and EDTA. Ethidium bromide. Agarose. Mix agarose powder with 100 milliliters of 1-xTAE in a microwavable flask. Microwave for 1 to 3 minutes until the agarose is completely dissolved. Pour the agarose into a gel tray with the well comb in place. Once solidified, place the agarose gel into the gel box, electrophoresis unit. Fill gel box with 1-xTAE until the gel is covered. Add loading buffer to each of your DNA samples in a separate sheet, mix properly and carefully load with a molecular weight ladder. Put the electrode lid correctly, red to red and black to black. Run the gel at 80 to 150 volts until the dye line is approximately 75 to 80% of the way down the gel. Turn off power, disconnect the electrodes from the power source, and then carefully remove the gel from the gel box. Using any device that has UV light, visualize your DNA fragments. The fragments of DNA are usually referred to as bands due to their appearance on the gel. Protocol for PCR. Prepare the reaction mixture in a sterile PCR tube. Mix and centrifuge. Gently mix the reaction mixture and then briefly centrifuge to collect the contents at the bottom of the tube. Initial denaturation, typically at 94 degrees Celsius for 3 to 5 minutes to denature the DNA. Cycling. Repeated cycles of denaturation, 92 to 95 degrees Celsius. Annealing, 50 to 70 degrees Celsius, depending on primer melting temperature, and extension, 72 degrees Celsius or higher. The number of cycles is usually between 30 to 40. The thermocycler cycles through these temperatures, allowing the primers to anneal to the template DNA and the polymerase to extend the primers, replicating the target DNA. The amplified DNA can then be used for sequencing. For restriction enzyme digestion, select restriction enzymes of gene interest set for primers. Determine an appropriate reaction buffer by reading the instructions for your enzyme. In a 1.5 ml tube, combine the following: DNA. Restriction enzymes buffer BSA, if recommended by manufacturer, BH2O up to total volume. Mix gently by pipetting. Incubate tube at appropriate temperature, usually 37 degrees Celsius, for 1 hour or for 16 hours. Always follow the manufacturer's instructions. To visualize the results of your digest, conduct gel electrophoresis.